Section 7.3 Improving Variant Classification Science. 7.3.1 Priorities for Improvement. Madhuri, Hegde. For germline, the top priority for updating the guidelines should be to integrate both the copy number variation guidelines and the ACMG sequence variant guidelines. We need to integrate biochemical cytogenetics and molecular genetics into the guidelines. For germline and somatic settings, a priority would be to utilize data on clinical responses to help establish whether a variant is pathogenic. There should be a feedback loop on the success or failure of precision therapies for individual patients. Third, we need a proper infrastructure for functional genomics. Additionally, in germline population-based analysis, we need to be watchful around benign variants, rare variant burden, and the evolution of polygenic risk scores. Also appropriate training is a grave unmet need in clinical genetics and genomics, as current training programs in molecular and cytogenetics that last just 2 years do not prepare candidates to contribute meaningfully in the clinic. Iain, Kerr. For loss-of-function genes in both germline and somatic settings, it is important to know the last known pathogenic variant that is supported well with evidence. That helps us understand how much can be deleted at the end of a gene before a pathogenic event occurs. Also, the use of in silico algorithms as a potentially deciding factor in classification has to stop. We need, and patients deserve, meaningful functional assays that can predict loss of function and lead to better medical management decision. Also, about ClinGen. ClinGen is largely an academic effort. The context of a high-throughput lab in biotech is not apparent to them. I applaud their efforts to reach some form of standardization, but they need to reach out to a wider community to truly affect some kind of change. Working successfully between the ClinGen committees and biotech can be unnecessarily challenging, and wider clinical genetics and genomics engagement could support overcoming these challenges. Stanley. We need a consensus on the correct transcript. Locus reference genomic is meant to be that database, but it only contains a fraction of genes. All variant curation should be based on the level of a variant by transcript by disease. Additionally, in germline specifically, we need a consensus on the incidence of diseases associated with genes, which would enable us to determine whether a variant's frequency is too high to be pathogenic. Elaine, Lyon. Understanding how reduced penetrance affects germline variant classification is important. What do we do with variants that partially disrupt but still retain some function? We can call those variants reduced penetrance variants or variants with different expressivities. I have used the term mild variants, but reduced penetrance is one thing the 2015 guidelines did not address. With reduced or mild penetrance, we need to think carefully about when such variants should go on a report. I would not call such variant classification inaccurate, but rather incomplete, because we simply do not know the potential effects. A lot of variants do something, but at what point do they cause disease? Eggington. Laboratories decrease their variant classification specificity when they use insufficient and/or low-quality data inputs for variant classification, and/or use overly permissive variant classification algorithms. Germline variant classification used to rely heavily on limited and unreplicated research databases, publications, and/or case reports. In time, these data sources proved to be unreliable. Many of these lines of evidence were demoted in the 2015 ACMG-AMP germline variant guidelines and are likely to be further demoted in future iterations. Therefore, the practice in germline of adding up weak lines of evidence to mimic a stronger line of evidence is inadvisable. In a concerning repeat of history, the 2017 AMP-ASCO American Society of Clinical Oncology and CAP Somatic Variant Classification Guidelines adopted weak lines of evidence within its Tier 2 category, likely in an effort to increase sensitivity, yet at the expense of increasing false positives. Clinicians use Tier 2 results to treat patients. However, the toll on patients of unnecessary treatment and the increasing costs of targeted oncology therapies provide strong incentives for decreasing false positives. Also, many laboratories overinterpret functional assay data in the literature, which leads to false positives. The decrease in activity or performance of a mutated protein or pre-mRNA splice site as measured by a research laboratory assay does not readily translate to human phenotype or drug response. Few assays are actually clinically validated for use in variant classification, and replication studies are rarely pursued. ClinGen's recommended approach to assessing functional assays is a great start, though I suspect we will ultimately need even stricter approaches. I believe, however, that it will be the continued development of large databases of quality-controlled sequencing with matched deep phenotyping of populations and drug response data that will be required for next-level variant classification science. 7.3.2 Relationship between research genetic testing and clinical genetic testing. Madhuri, Hegde. Variants in genes are being identified all the time, though many do not have enough evidence for disease or disorder causality. It is unfortunate that laboratories are beginning to report variants as causative of a disease or disorder when causality is not apparent and is not supported by sufficient evidence. We need clinical research that accompanies basic research. What we are trying to do is understand the functionality of our basic biological systems, and we need basic researchers to have robust collaborations with the clinical community, with research funding mechanisms that encourage these collaborations. Elaine, Lyon. Submissions from ClinVar coming from both research and clinical labs is a double-edged sword. If I encounter a discrepancy in ClinVar, then I need to go in and look at it. In my experience, often that discrepancy is coming from a research lab. To be clear, I want to know if a research lab identified a variant and if they have evidence of pathogenicity. However, I would appreciate improved transparency around whether a submission is research or clinically based in origin.